my name is Jessica Martin. I'm a second-year master's student in the Galapagos Research Lab at San Francisco State University. My research focuses on the genetics of the Galapagos endemic lava gull. The Galapagos Islands are located 1,000 kilometers off the coast of Ecuador in South America. Because of their isolation, they host a high proportion of endemic species. These are species that can only be found in the Galapagos Islands. For example, there have been 199 bird species recorded in the archipelago. 47 of them are considered endemic. One of these Galapagos endemic species, the lava gull, is the rarest species of gull in the world. And though we don't know exactly how many there are, the most recent estimate has that there's between 300 to 600 individuals. These gulls are scavengers and predators, often drawn to urban areas because of the high availability of human food and waste. However, their proximity to these environments really increases their vulnerability to anthropogenic disturbances and pollutants. Due to their small population size, very little genetic research has been conducted on this species, thus limiting our understanding of its population structure, evolutionary history, and conservation status. My work aims to sequence and assemble a reference genome for the lava gull. In July of 2024, we went to the Galapagos and we collected a blood sample from an adult female lava gull on Santa Cruz Island and brought this to the Galapagos Science Center on San Cristobal Island, where we performed DNA extractions, library preparation, and sequencing using an Oxford Nanopore Technologies PromethION 2 Solo, which is a portable sequencer that allowed us to sequence the genome of the species on site in the Galapagos. From there, we returned to San Francisco with the raw reads that we sequenced there, and we conducted all the bioinformatics work at the California Academy of Sciences, including assembling the genome with the assembler fly. The manuscript of this work was published in the journal G3, Genes, Genomes, and Genomics at the end of December. I am currently working on conducting population genetic analyses through a collaboration with a PhD student based in Australia. For this, we are using low-coverage whole genome sequences from 32 individuals across 4 islands to estimate effective population size, assess population subdivision, identify distinct units of conservation, and evaluate levels of inbreeding. These high-resolution genomic analyses provide critical insight into the evolutionary history and current conservation status of this imperiled species and offer valuable guidance for management and protection efforts. I want to say thank you to my lab, my advisors, ARCS, and everyone who has helped me and been with me on the journey of getting my master's and my entry into science. Thank you all so much. This work would not be possible without you.